Now let's take a look at how trichromatic theory and opponent process theory say that these two processes, these two ways of perceiving color interact, because they have to come together. And what we know is that first the cones absorb different wavelengths of light, right? So the short wavelength cones, medium wavelength cones, and long wavelength cones, they each specialize in absorbing their own range of wavelengths of light. As a result, the cones produce a unique response between the short, medium, and long wavelengths, so that as the wavelength changes, the color changes, the cones in the retina produce a unique pattern of neural activity. Once that happens, once the cones do their thing and create their unique pattern of neural activity, once that happens, then other neurons, for example, bipolar cells in the retina, will react differently to stimulation by, say, long wavelength cones versus medium wavelength cones. So for example, a bipolar cell might be excited by long wavelength cones but inhibited by medium wavelength cones. And so let's take this example. Imagine the color is color number 1, and it's this wavelength. This particular wavelength is more towards the middle, and it's going to activate the medium wavelength cone more than the long wavelength cone. So we see that pattern of activity here where the medium cone is more active than the long wavelength cone. And then as a result, this bipolar cell that is inhibited by medium wavelength cones is going to get more inhibition from the medium cone than it's going to get excitation from the long cone, because the medium cone is stronger. And so inhibition will rule the day here for this particular color, and this bipolar cell, this opponent process neuron, will become inhibited, and as a result we perceive the color green. In contrast, for color number 2 is a longer wavelength color. It's going to activate the long wavelength cone more than the medium wavelength cone, and we see that here. As a result, that bipolar neuron that is excited by the long cone but inhibited by the medium cone is going to become excited. Why? It's going to get a net amount of excitation. There's more excitation than inhibition because the long cone is more active than the medium cone. And so we see that net excitation here. This neuron becomes more active to color number 2, whereas it became inhibited to color number 1. And when this neuron becomes more active, becomes excited, the rest of the brain interprets that color as red. And the idea is that the opponent process decreases any ambiguity in the neural signal for color. So it's either red or green. It gets rid of any possible ambiguity there. Now let's see how this works. Opponent process neurons are found in the retina. That starts with the bipolar cells and the ganglion cells, but they're also seen in other areas of the brain like the LGN and the visual cortex. We can see here how the system works for the different channels. For the red-green system, the ganglion cell, for example, compares the activity of the medium and long wavelength cones. That's exactly what we saw here. For the yellow-blue system, then the ganglion cell would compare the activity of the short cone with the combined activity of the medium and long wavelength cone. So in this case, it's short cone versus the combined activity of medium and long. If the short cone is more active than the medium and long are, then this system would signal that the color is blue. But if there's less activity in the short cone and more activity between the medium and long cones, then the system would signal that the color is yellow. So we get a nice clear distinction between blue versus yellow here because of that. So it works just like the red-green system, except it's combining the activity of medium and long cones. Now that we understand this, we can also take a look at color deficiency. This is sometimes popularly called color blindness, but it's not really being colorblind, is it? It's just a difficulty distinguishing between colors. And it's normally easily identified using what are called Ishihara plates. And if you have normal color vision, the number 12 should sort of be obvious to you, clearly visible. But in some cases of color blindness, the pattern created here by the different colors will be harder to see because the visual system is not distinguishing between these two colors. There are three types of color vision deficiency depending on which of the color perceiving cones is missing. There's protonopia, and individuals would be considered protonopes if they are missing the long wavelength cone. In that case, these pictures, as shown here, are how they would be seen by a person with normal color vision, and a protonope would look like this. Notice that deuteranopes who are missing the medium cone have very similar perception. This is generally referred to as red-green color blindness. So whether you're missing the long wavelength cone or the medium wavelength cone, the effect is really similar. And it's the most common type of color blindness, red-green. People missing the short wavelength cone have something called Tritonopia, and they might be considered Tritonopes. And their perception is more like this, where they see reds and greens, but really no blues and yellows. Now I want to point out that in red-green color blindness, you're missing both colors, right? Even though the individual is only missing one type of cone. Likewise, if the person's missing the short cone, they're blue-yellow colorblind. The reason for that is without the proper input from the cones, the channels can't work properly. So in Tritonopia, the blue-yellow channel can't work, but the red-green one works just fine. And in red-green color blindness with protonopes and deuteranopes, it's the red-green system that doesn't work properly. So the opponent process is not able to function because it's missing the input from either the long cone or the medium cone. Here we can see the spectrum as visible to an individual lacking the long wavelength cone or the medium wavelength cone. So blues and yellows are detected, but really not much in the range of reds and greens. So it's not that they don't see the colors, it's just that they have a harder time distinguishing between the colors. And here we see blue-yellow color blindness in an individual missing the short wavelength cone. These arrows point to what's called the neutral point. That is the location where their two functioning cones overlap and create an equal amount of activity, which the brain confuses with a gray color. So for example, in this individual missing a long wavelength cone, they might have some pattern of response like this to the short cone and a pattern like this in the medium cone. And so this part right here, this 492, is where the two cones are equally active. I'm sorry, I drew it very poorly, but it's where the X is here. It's where these two curves cross perfectly. And so this wavelength of light produces equal activation in both the short cone and the medium cone, which the brain would normally interpret as white or a shade of gray, right? Likewise, there's the neutral point here for the individual when where the short and long cone overlap, and here where the medium and long cone overlap.